mynews.studio.
Surgeons are now handing over living brain tissue, and for the first time, scientists are able to watch Alzheimer's actually begin. Could this change how we understand the disease?
Today's date is 2025, June 8th. Let's dive in. Scientists have used fresh human brain tissue from neurosurgeries to recreate early Alzheimer's stages in the lab. But why is this so important? Until now, research mostly used animal models or tissue from people after death. Those don't show the real-time responses of living human brain cells.
Right. They took small pieces of brain tissue, kept alive in a dish for weeks, and exposed them to amyloid beta. That's the protein widely thought to trigger Alzheimer's. But here's what stood out. Only the toxic form of amyloid beta caused lasting damage to the synapses. The tissue just couldn't repair itself.
Just to make sure I'm following, synapses are the connections between brain cells, correct? And in Alzheimer's, those connections break down, which leads to memory loss and cognitive issues.
Exactly. What the researchers saw was that even small imbalances in amyloid beta could disrupt those connections. And when they worked with tissue from the temporal lobe, which is one of the first regions affected in Alzheimer's, the tissue released higher levels of tau protein. Tau is another major factor in the disease.
It seems pretty significant they could track these changes as they happened. Is this really the first time researchers have watched brain tissue deteriorate in this way, outside of a living person?
Yes, this method is new. It lets them observe minute by minute how the disease progresses. It's a much more accurate model of what's happening in the human brain, and it means experimental drugs can be tested on living human tissue before any clinical trials start.
That could really speed up drug development. If you can see whether a drug protects synapses in real tissue before testing it in patients, that's a big step. Do you think this could lead to new treatments quickly?
That's the question everyone's asking. The researchers are hopeful. By modeling early Alzheimer's more accurately, they might identify drugs that work in human brain tissue. That could move things from the lab to patient care much faster.
And this approach, it isn't limited to Alzheimer's, is it? If you can keep living brain tissue alive in the lab, could this work for other diseases like Parkinson's or ALS?
That's already being considered. The same approach could let scientists study early changes in a range of disorders, not just Alzheimer's. It really opens up new possibilities in neuroscience.
The need is huge. Over 55 million people worldwide have Alzheimer's, and the impact on families and healthcare systems is massive.
Absolutely. This breakthrough gives hope, not just for understanding the disease, but for finding treatments that actually work, and maybe, in the long run, a cure.
So, let's sum up. For the first time, we can see Alzheimer's begin in living human brain tissue. We know which proteins cause the damage. We can watch the process, test drugs on real samples, and maybe speed up the path to new treatments. And there's potential to use this for other diseases too.
That's it. It's a turning point in research, and there's real hope now for millions of people around the world.
Thanks for joining us for this conversation. We'll keep following the science as it develops.
Thank you for listening. This episode was powered by mynews.studio. Try it now for free.